everyone. I'm so sorry that I can't make it there in person today, but the good news is I can show you the lab and you can see what a day in my shoes is like. What I conduct is exercise-based virtual reality interventions, specifically using functional near-infrared spectroscopy, also known as fNIRS. And what we're going to be focusing on is methods in developmental cognitive neuroscience research, specifically how we can account for different hair phenotypes. Assisting today is also a close colleague and co-author on this work, Suanna.
Hi, everyone.
The first thing that's really important is head circumference. So prior to participants coming in, we show them a video, but they send us their head circumference, and this is how we know what cap size to use. And this is really important because the cap should be pretty snug on their heads, but not uncomfortable. These are spring-loaded caps, and these go over the sources and detectors. So in our cap accessory box, what you'll see is it ranges from a 0 to a 3. The zeros, they have no springs in them. The tension 3s that we use, that is the amount of, tension that's put on these sources and detectors to keep them in place. For these specific studies, we've used tension 3s all around the head, and that makes sure that the sources and detectors stay really close to the scalp. These are the products that are recommended to use, and after a year of pilot testing, these are the products we've learned that you actually need to use. So on the left side of Suanna's head, I'm going to use the more traditional methods that have been used in fNIRS research. And on the right side of her head, I'm going to show you the methods that we've used in the lab to account for different types of hair. First, we always ask participants, is it okay if I touch your hair prior to capping?
Yes.
Okay, so we always ask consent because consent is important. We usually give them a middle part, which is great because that's in right now. And also Suanna has a middle part, so it's kind of easier. Using the traditional comb, you kind of part here, but because Suanna has really thick and wavy hair, the comb is basically, it's getting a little tangled. We don't want to do that. So something that we do is we order these disposable combs, and what these are called from all the barbers that I've consulted with are rat tail. They have rat tail ends. And so what this can do is for all different types of hair textures is it very easily just slides through Suanna's hair. And what this is doing is you are parting the hair evenly across the head, and this makes sure that it's kind of like evenly distributed so it's not all bunched up, which makes it really hard to get good signals underneath all the optons. I learned consulting from other researchers doing this type of work, and also going to black hairstylists and black barbers in the San Francisco area, is when you have really coarse hair, you never brush it dry. Why? Because it breaks easily, and also you can't put a comb through it. So we spray it with water. So I'm just going to spray Suanna, so you cover the face, and I'm just spraying Suanna's hair lightly with water. The second thing is a wide tooth comb. So this is appropriate for hair that is wavy and curlier than straight hair. I'm just going to comb Suanna's hair, and you can see that it just really easily goes through her hair. And I'm going to show you the traditional comb, and if I brush through Suanna's hair, this is about the farthest it's going to get. It kind of just gets stuck there. So I'm just going to remove that. What is this, you ask? So this little comb here, that takes into account people with bangs or these little baby hairs here. So what we do is we kind of comb these back like this. Okay, so what we're going to do is we're going to run calibration on Suanna's head, and before any sort of testing, this is really important because you typically want something in the acceptable or excellent range. If any channels are red, that means that there's something obstructing the source and detector channel, and it's typically hair or cables. So in traditional fNIRS labs and also used in EEG labs, you use the end of Q-tips to kind of move any hair out of the way, and also this hypoallergenic gel. In our lab, what we found has been useful for keeping coarse or more curlier hair out of the way is braid gel. So this is what we use, and I'm just going to really quickly just show you how that's done. And so you can just see here from Savannah's head from the start is the right side, there's more green compared to the left side, solely from combing the hair. It has been 30 minutes because that is our timing on how long we can actually spend, because we know that working with children and with clinical populations, you can't spend forever on the pre-capping process because of fatigue effects. This is when we call it, and that's basically like, this is the best that we can do. So on the right hemisphere, what you can see is that with the braid gel and the combing and the water spraying, we have almost a perfect, acceptable, excellent quality channels, minus one channel on the parietal lobe. On the left side, where we use the traditional Q-tips and also the hypoallergenic gel, we have 4 bad channels. And you can see here that even if I take out a sensor and I move Savannah's hair around with the back of the Q-tip and I load it up with gel here, that it's not budging, it remains red. So there is your live presentation of how we can optimize the signal quality for diverse hair textures.
Hi everyone, thank you so much for watching my talk live demo on how we can increase inclusivity and equity in cognitive developmental neuroscience research using fNIRS. So just a very quick overview. fNIRS measures changes in blood hemoglobin concentration by imaging the brain with low levels of light. It's non-invasive, it permits mobility, it's not confined to a small space and loud environment, it's less sensitive to motion artifacts, and it's relatively portable and inexpensive. And you'll notice that the articles have nearly doubled since a decade ago. NIRS uses the same technology as pulse oximeters, which most of you have probably seen at doctors' offices. One light source, one detector that leads to a channel. Franz Jobst in 1977 was actually the first to prove that near-infrared light can actually penetrate through the skull and biological tissues to the cortex. So what this montage shows you is a prefrontal cortex layout, except unlike the pulse oximeter of one source and one detector, there's 4 sources and 8 detectors to give 10 channels on the prefrontal cortex. Most of my prior work has shown that with 4 to 5-year-old children, that resting state functional connectivity using fNIRS has high test-pretest reliability, and it also predicts later executive function through a longitudinal study. Now, this is relevant because in my prior work at Carnegie Mellon, I used a tech-N machine, laser sources. And you know, when they say not to shine lasers, like these pointer things in your eyeballs, the same risk comes with optical fibers. So when I came to Stanford, I was exposed to this advanced fNIRS technology using LED sources, no risk to eyesight, lightweight and compact, actually portable, more optodes. And instead of a prefrontal cortex montage, I have a frontoparietal montage. Easy, right? There should be no challenges here. But then, yes, I did find challenges. So I think this is one of the first times I scanned my own head, and you can see that what these red channels indicate is that they are very noisy. This red is an example of a perfect fNIRS signal showing that hemodynamic response. This blue is a noisy signal, and that typically indicates hair. Even with the standardized methods to optimize NIRS signals, such as the ones I showed you in the video, like gel and Q-tips, has significant challenges for darker and curlier hair types. My collaborator on this work, Dr. Jasmine, who is a biomedical and electrical computer engineer at Carnegie Mellon's Neuroscience Institute, one of her most recent systematic reviews showed that only 1% to 2% of the top biomedical optics and human neuroscience journals using fNIRS with human subjects report race and ethnicity, and only 3 to 7 report hair types. In my work using fNIRS, I report race and ethnicity. And I hope that this becomes a more common practice among developmental researchers. So the data that I'm going to show you is from 54 high school and college students ages 17 to 21 recruited from the local community in the Bay Area. So my earlier work was with preschool age children, but my current work is focusing on a population consisting of a developmental group of adolescents and young adults experiencing attentional difficulties who are often overlooked. This is their demographic breakdown and also their ethnicity breakdown. Through collaboration with Jasmine and consultation with the dermatology department, which is way outside my domain, we used established scales of hair color and curliness. So prior to being capped, photographs of participants' hair using a 64 megapixel camera are taken in the same lighting, room, and position. Then 2 research assistants subsequently coded the hair phenotypes with inter-rater reliability of 0.91 for color and 0.85 for curliness, indicating substantial coder consistency. I also have this nice database of hair now. Some of the most innovative practices, such as researchers like Jasmine have spearheaded, are 3D printing electrodes for curly and coarser hair, and also compensation for participants to go to salons and barbers to get their hair braided. There are some practical constraints. For example, what if you're a postdoc with limited discretionary funding and don't have large grant money yet? So I actually went to Black hairstylists and Black barbers in the Bay Area, mostly located in the San Francisco region. Importantly, I talked to these hair cosmetologists about appropriate norms, tools, and products, which are the products you saw in the demo video you were showed earlier. What I found from this data is that with these methods, we have 83 to 100% fNIRS scan quality. That's the range. So omitting 5 channels at most, and with an average of fNIRS scan quality of 94.06% or a 5.94% exclusion rate using these methods that you saw earlier. In summary, this study provides a cost-effective and easy-to-scale fNIRS practice to optimize data acquisition for diverse hair types and textures. It established reliability of standard hair phenotype scales in developmental cognitive psychology research. It investigated fNIRS signal quality and hair phenotypes associations, and it also reported demographic factors to increase inclusivity and representation of diverse sample population. Now, addressing these concerns is not merely a technological challenge, but carries profound sociocultural implications. If unaddressed, there's a risk of reinforcing biases against specific demographic groups, notably those with coarser and thicker hair, furthering widening the existing representation gaps. And finally, I would just like to thank the Army that made this research possible, NIH and Stanford's Trailblazing Trainee Award Program, the Wuxi Neuroscience Institute, and the BioX for funding this research. And I also wanted to thank the organizers, Tara and Jasmine, for organizing this pre-conference. And again, I'm so sorry I can't make it there in person. I have serious FOMO.